Good afternoon, everyone. I extend my warm welcome again to you all. We will now be proceeding to our next event. We are pleased to have Dr. Premalatha ma'am with us. She will enlighten us regarding algae, prospects of algae as a fuel and value-added materials. She is our head of department. She has more than 15 years of academic and research experience in areas of energy audit, energy efficiency, algae, and CO2 sequestration. She is serving as a distinguished author for many high impact research articles and rendering consultancy services for various educational institutions.
Good afternoon. This is on prospecting of algae, sustainable fuel, food production, and wastewater treatment. Uh, I had also listened to Dr. Reena, who spoke just previous to this lecture on the algae. So this will be a continuation of her lecture. Uh, Hopefully, not much will not be repeated, should not be repeated. Uh, So this lecture, I would like to give the maximum information what on our experience with algae on what are the possible things that could be done in an economical way of taking algae to the next level. What is our experience? That is what I would like to share. So starting with the applications of algae, I would like to cover one with reference to application of CO2 sequestration. What is the potential of algae to sequester CO2? Because all of us know we are running on a climate change uh, problems due to which we have a lot of issues related to environment, health, agriculture, everything, and with other products. Also the food production in line with the utilization of waste as a feed material to the algal growth, how food production can be enhanced, and the role of algae on wastewater treatment. So these are the aspects that I would like to cover in the this lecture. So this slide is again I said in the morning there is a problem of CO2 in the atmosphere. It keeps increasing. The CO2 keeps increasing in the atmosphere. Now the current level of CO2 is gone 110 plus ppm. So the information available now says if it is touching 2 ppm, it is rising with the 2 ppm per year. The level of rise, rate of rise is 2 ppm of carbon dioxide per year is increasing. If the CO2 level touches 450 ppm, then we will have the situation or deterioration in the atmosphere will become uncontrollable. So this is the status that we need to take kind of steps so that the carbon emissions into the atmosphere is getting reduced. So whatever the problems that we face today due to pandemic, the problems that are expected due to the impacts of climate change will be much higher than what are the problems that we are facing. So as per the calculation, if it is now 412 ppm, remaining is 38 ppm. So 38 divided by 2, you have 19 years. Within 19 years, some fast response has to be taken from the emission side and control side, so that the environment CO2 ppm is sustained in the atmosphere, so the impacts become less. So in this aspect, there's a lot of things that could be done by the algae. That is the first aspect of the content that we are going to see. So this previous slide also says, generally previously people said it is 2 ppm per year was the rise. But this data, if you just see, it is becoming 2.5 plus or minus 0.1 from 2017 and 2.2. It rose from 2 to 2.2 in 2016 and 2.2 to 2.5 in 2017 as per the reference. So it is very alarming that everybody needs to worry about the amount of CO2 that is getting into the atmosphere, so that there is a possibility of sustainability that we will be able to create in the atmosphere to live further. So this is a kind of data that I took. Generally, we think CO2 is very inert gas, so even 19 years is too far, so that we can sit comfortably or something like that. This is some kind of data that I took. Even CO2, if the concentration goes beyond certain level, what it can happen. So it talks about 8 hours threshold limit is 5000 ppm. Uh, if you just see inside the room, if you are sitting maybe 100% or 200% without proper number of air changes per hour inside the room, the CO2 concentration easily goes beyond 1500, 2000 ppm. So if you are sitting in 5000 ppm for 8 hours, that will create a large amount of impact in a shorter span of time. And if you are exposed for 30,000 ppm, which is 3% of ppm, 3% of carbon dioxide for 10 minutes, it is again a problem. 40,000 or 4% of CO2 is detrimental to the life. So this is the information. So the amount of CO2 needs to be controlled and uh, reduced into the atmosphere. That is what I said in today's morning welcome address. One point I said, it is something called the circular economy and circular bioeconomy. What is the difference between these two? Circular economy is recycling or reducing something into the atmosphere. So when you recycle or reduce something, the usage of something, then what does it, how does it have an impact on the environment? It is actually you are reducing that much amount of CO2 emitted into the atmosphere because you are not consuming fresh material. Since you are reducing the fresh material being consumed, the amount of CO2 entering into the atmosphere is reduced. That is a concept of circular economy. So there's a possibility you do a lot of things with the metals and minerals on these aspects. Whereas the same concept when you extend for biological system, bioeconomy, circular bioeconomy. So in this case, what is done with reference to carbon dioxide is you are introducing new renewable carbon into the atmosphere. There you are reducing the CO2, fresh CO2 entering into the atmosphere. Here you are introducing new renewable carbon into the atmosphere. So coal itself, we call it is non-renewable because it takes many thousands of years to get it done. If you are able to produce the same carbon, coal or carbon, the carbon which is a fuel in the coal, in a very faster manner, then it becomes renewable carbon. So if you are able to produce a renewable carbon and introducing into the usage, then also you will get a better sustainable environment. So this concept we call this is a circular bioeconomy. See how far it is possible to reuse or recycle or get more products or cascading processes possible with reference to bioprocesses is a challenge. So what to be done with reference to that? So anyhow, these either circular economy or circular bioeconomy has to lead to reduction in carbon into the atmosphere so that the environment becomes safe to live. So here we talk about algae. As Dr. Reena said in the morning, it is only a photosynthesis reaction which captures the light from the sun, and the same thing whatever happens in the terrestrial plant. The only difference is the rate of growth or the absorption of CO2 becomes higher than a normal terrestrial plant. So that is the difference between the terrestrial plant and algae. It sequesters more. The carbon dioxide, it is sequestered, it is split into carbon and oxygen and H2O. So it becomes a CH2O n times molecule. So any species of algae fixes the carbon dioxide in the form of CH2O n times molecule. So this molecule could be either in the form of protein or carbohydrate or lipid. So based on the type of species, the amount of protein, lipid, carbohydrates fixed by the species will be different, and fixed by different atmospheric conditions will be different, fixed by in the presence of different nutrient conditions will be different. So the fraction of lipid or protein or carbohydrate is purely a function of the type of species and the type of operating conditions that are used. Here we call this operating conditions is the amount of CO2, the amount of light, the amount of nutrients. You call that as the temperature. So all these things become operating conditions. So the carbohydrate, lipid, protein fraction is a function of the type of species. the operating parameters. Uh, so the operating parameters, when I say it is operating parameters, that varies from place to place, location to location. So for a given location, you have to choose an appropriate species so that it gives you whatever the product that you are interested in producing. If you are interested in producing a protein, so you select a particular species, or carbohydrate, a particular species, or lipid. There are certain species which can yield for the same conditions. That particular species will convert the CO2 light into more amount of lipid. So based on the uh, type of species and type of the operating conditions available in a location and the type of reactor, also you will have a different type of percentage fractions that will be fixed by the species in the form of carbohydrate, protein or fat. So this is the point of station. As I said, it is renewable carbon. So why do we call it as renewable carbon? Because the CO2, whichever is consumed by the photosynthesis, same amount of CO2 is getting emitted into the atmosphere when it is burnt as a fuel. So the same thing can be recycled back. So it becomes a renewable. So why biological sequestration? Compared to other types of sequestration of carbon dioxide, maybe chemical or geosequestration or ocean sequestration, all these things, the biological sequestration is a permanent solution. It is a permanent solution. So once the carbon is fixed in the form of carbohydrate, it's not going to get reversed back, or protein, fat, everything. You are able to get a product. You are able to fix the carbon dioxide in the form of directly as a product. Let us say if you are taking carbon dioxide, you pressurize and store it somewhere, then you take it for the transportation, compression, then transportation and storage cost. Then you utilize as a chemical production of chemicals. You can use carbon dioxide. So all these things compared to that, this place, if you make a life cycle analysis of compared to other processes, it can be done. So this biological sequestration is on the place where the CO2 is available, where the light is available. So you are not going to, it is not required to purify the CO2 to the 100% purification level, then you apply the CO2, not required. Even 5% CO2, which is emitted from the industry, or 15% CO2. The flue gas emission has got 15% of CO2 in its emission. So it is not required that you have to make 100% pure CO2 gas, then you supply for algal growth. Nothing like that. Because as we know that algae grows with the atmospheric CO2 concentration. So which means that atmospheric CO2 available in the atmosphere is only 0.04 percentage. So with that also it can grow, and you supply an industrial pollutant with a higher percentage of CO2 also it grows. So which means that you don't have to separate a pure CO2, then compress, then you have to supply, nothing like that. You have to consider the temperature at which the gas is available. Then what is the concentration at which the CO2 is available in the pollution. So if the species is able to tolerate that high concentration of CO2, then directly the pollutant gas can be supplied for the algal growth at the right temperature. So this is so there are no issues with reference to all these aspects which are there in the other types of capture and storage mechanism, and directly it converts the carbon dioxide into biochemical composition. So then you can go with the additional production of other chemicals from the biochemicals which are produced. So there's so many advantages are there with the biological sequestration. So the carbon sequestration by algae is very much meaningful. Only problem is it is a slow process. So for that, what we done, and or the problem with the slow process is providing higher amount of area. What is to be done? That we'll see now. So here, carbon sequestration by algae is given as a renewable fuel. That is what I said. It is circular bioeconomy. You think of going for renewable carbon being produced from the biomass, then you supply that to the vehicles. So this, if you just see, these are the points that I said just now. So the what are the variables when you talk about carbon sequestration by algae? What are the variables? First variable is, as I said, it is algae. the type of species. So this slide gives you what is the potential of different species in terms of CO2 getting sequestered. So based on the species, the amount of CO2 sequestered varies. So once you fix the type of species, then the other variables include percentage of CO2 at the influent. So that is where I said that it is different percentages of CO2 concentrations were studied by different authors. So we have also studied up to 100% CO2 being supplied to the algal species. It is not with the objective of separating the from the industrial pollutant and supplying, but not with that as an objective. The objective was there are certain industries which produce CO2 at 100% percentage purity level. For example, it is distilleries where the fermentation of molasses happens to produce alcohol. To produce alcohol, it is the fermentation section is 100%, the 99% of CO2 is coming out. So there are other industries, fertilizer industries and other industries where there is 100% CO2 is also emitted. So we also carried out a study with reference to applying 100% CO2 from the cylinder. Then we checked with the growth of algae. There are certain species which can grow at higher 100% of CO2. Also, it's not all the species. So, as Dr. Reena said today in the morning, it is going to be a cyclic process. What is the amount of light available, amount of CO2 available, amount of nutrients available? So, if you have, with reference to India, the amount of light availability is very huge. The light is measured in terms of micromole per second per meter square. Out of the entire solar spectrum, you will be using only the visible spectrum for photosynthesis, which is available from 400 to 700 nanometer, which is equal to 45% of solar radiation. So this only can be taken by the species for its growth. So you have a loss in efficiency there itself. So there's a limitation that we need to understand why the photosynthetic efficiency is very low. Because it cannot take up the wavelength, the radiation available in the entire spectrum of the entire solar spectrum. So you have a visible radiation which is measured in terms of micromole per second per meter square. So we measured here up to even 3,000, 4,000 micromole per second per meter square also. So what I mean to say is you have a large amount of radiation available, large percentage of CO2 also available from the pollutants. And if you have a nutrient supply from marine water, let us say, then absolutely the cost of cultivation goes down to drastical levels is a possibility. So we have worked with 100% CO2 also, it is able to grow. So then it is flow rate. Flow rate is a variable. Why the percentage of CO2 is limiting in certain species? Why the percentage of CO2 is limited in certain species? The point is when you supply more the concentrated CO2 or more quantity of flow rate. The next column is flow rate. When you supply more amount of flow rate of CO2, again the pH of the medium will come down. So all of us know that any biological process, the pH is part of the process. So pH has to be maintained for a particular species at the particular level. If it is not maintained, the entire process will be stopped. So due to which you need to have a control on percentage of CO2 and also flow rate of CO2. Then the mode of operation, whether it's a batch or a continuous operation. Then the type of reactor. So there are two types of reactor, basically speaking, as Dr. Reena said, it's an open pond and a closed photobioreactor. So people have studied about different types of closed photobioreactor and they've given what is the yield that they are getting. So then these are the variables and the next four columns are all the output. What is the output? So if you vary percentage of CO2, flow rate and mode of operation and type of reactor, what is the amount of CO2 sequestered? So what we need to understand here is as you vary all these parameters, either the type of reactor or the percentage of CO2, all these things has an impact on amount of CO2 sequestered per hour. So this is what we need to understand. And the productivity of the species again is going to be different with reference to each species and the type of reactor and operating parameters. So, as Dr. Reena said, we need to have something as a benchmark to be achieved. Let us say, in literature, people have reported on 50 grams per meter square per day, and we should aim at least 100 grams per meter square per day. So, like that, if I want to change the yield to 100 instead of 50, the same area, then what I need to concentrate, either the species or the reactor or the operating parameters. So all these things need to be considered so that the yield becomes better. So ultimately, as you increase the yield, then the system becomes economical and also the land area becomes small. So this, then again, when we talk about the algae, why we are going with the fuel as an application, as we all know that we talk about first generation, second generation, third generation fuel. This again, previously people used food material for the fuel, and second, bio resources, bio residues were used in the second generation, and third generation we can cultivate microalgae and use it as a fuel. Again, when you talk about microalgae, if you again if you produce only fuel, it may not become economical immediately as of now. Instead, you can think of more number of products other than only other than instead of only looking the microalgae as a fuel feedstock. So let us say you think of producing fuel, and from the residual material you try to have a kind of other value added products that I will be discussing in the later slides. Then the system becomes economically viable. So these are the things that we are looking as why again because of the environmental friendly nature. So we are going with that. So the types of algal species you can have a unicellular and multicellular filamentous. It varies with reference to its shape, and 2 cells or 4 cells or 8 cells, the way in which it is available. These are the classification. So versatility, again, what are the advantages of growing algae is, it is not that it can grow only in fresh water, it can grow in marine water and also wastewater. So that is where I also said that we will also take wastewater as one of share our experience on wastewater, growing algae in wastewater also. So it does not need an exclusive kind of resources for its growth. That we need to understand. So either it is other than CO2 and light, the only costly input required for algae is water and nutrients. If the water and nutrients are available either in marine water or in wastewater, then totally the cost of cultivation becomes it can be drastically brought down, as I said earlier. So uh, these are the products that can be possibly obtained from the algae. This is what I said. Instead of one product, you have to think of more number of products being produced from the algae. Then it becomes economically viable. Then the overall process becomes economically viable. Instead of only algae to be used as a fuel, if you are able to use that residue either as a biofertilizer or as a feedstock for an animals, then it becomes the economical process. So here, what I would like to insist here is, let us say you go with wastewater treatment and CO2 sequestration. So you get two advantages in this. One is the nutrients available in the wastewater is going to be used as the pollutant available in wastewater is going to be used as nutrients for the algal growth, and CO2 is going to get sequestered from the atmosphere. So you will be touching upon the water pollution as well as air pollution in the growth conditions. And once you get the biomass out of it, then if you are making a value-added products, then the economy becomes very good. So this is again the comparison between why microalgae. You have a better photosynthetic efficiency in microalgae compared to higher plants. Here it is given as 9%, higher plants 6%. Again, it is a very high efficiency. What is being given here? You still get a very low efficiency for higher plants, maybe 2%, and 6% or 7% for photosynthetic efficiency. Maximum theoretical efficiency for microalgae itself is only 12%. The reason is you have, as I said, only 45% of solar input is used, visible spectrum alone is used. Out of that, again, if the radiation intensity is very high, again you have a loss due to photosaturation and photoinhibition effect of microorganisms. So due to which your photosynthetic efficiency further comes down. So all photo-related reactions are very sensitive to the temperatures. Again, due to the temperature, the algal system will not grow good. So these are the reasons why the efficiency of algae is very less compared to other conversion efficiencies. So here you have a good amount of carbon uptake for the same area, the same volume. If you just see, it is 25.6 compared to 0.9, less than a kg of carbon per trees. And water requirement is again less. And again, instead of water, you can think of going with wastewater or a saline water. So what needs to be fixed is first the water quality. What is the water quality available in a given location? So if it's a wastewater or even a water quality you want to use, so that has to be fixed. So in that quality of water, which type of species can be grown the good growth rate. So this the approach has to be in that aspect. It is not that selecting a species then I grow with this. You study the environmental conditions properly, then you take a species which can get easily acclimatized with the given environmental conditions. It has to be from back to the front. It is not developing a strain in the lab, then taking it to the outside. It will take a longer time and it will sometimes it may fail. So you have to, with reference to biological system, the chemical processes, when we talk about scaling up, we do in the lab scale, then pilot scale, then it has to go to the industrial scale. Whereas in my opinion, the biological processes should be in the reverse direction. So you have to do it in the site. Then what are the problems that you face in the site? That has to be studied in the lab scale. Say, let us say, for example, high temperature is an issue. If that is the case, outside, if you face a higher temperature, then you have to cultivate the algae at the lab scale at 40 degrees centigrade, not 20 degrees centigrade or 30. So what needs to be studied is you have to study the environmental conditions and water quality available. You take that conditions into the lab, then you study and acclimatize the species, then take it outside. So it has to be done like that. So we also have a shade facility where we grow the algae in a large scale. So these are the advantages of algae. It fixes a huge amount of CO2. And when we talk about the conversion process, algae can be converted chemical, biochemical, and thermochemical conversion. When we talk about chemical conversion, where we talk about a lot of product being produced, one such product is again fuel as a product. Biochemical conversion, as I said, it is. If you talk, this slide is only talking about the fuels. Let us say if you are interested in getting only fuel out of the biomass, then what are the routes available? So you can choose a chemical route or biochemical route or a thermochemical conversion. Chemical conversion, it is transesterification. Then you get a biodiesel. Again, in transesterification, it has lot needs to be innovated so that it becomes better net energy. It becomes less for in terms of chemical being put into the process. It has to be reduced. So, and biochemical conversion, you can get either ethanol or biogas anaerobic digestion. Through fermentation, you get bioethanol. Thermochemical conversion, you get bio-oil, biochar, or syngas through pyrolysis, gasification, or hydrothermal liquefaction processes. So if you are talking about only fuel, so then these are the processes available to get the fuel. So you get algal biodiesel out of the, you get first lipid, then to go with the transesterification process to produce biodiesel. So again, biodiesel, you have to think of in lipid, what is the kind of fatty acids available? You have different species having different types of saturated and unsaturated fatty acids available. So that you have to check what is the quality of the biodiesel that you are interested in terms of fuel aspect. What is the calorific value? What is the core point? What is the flash point? What is the cloud point? These are the things that you need to define. So if you want to attain these, either you want the species to or the lipid to contain more amount of saturated fatty acids or unsaturated fatty acids. So this has to be taken care. So in general, if you have both in almost equal quantity, then it is good. Whereas the other aspect is, even though we cultivate algae for fuel purpose, then there will be a question. If it's a polyunsaturated fatty acids, then it has a lot of other food values also. So here it gives a comparison on the oil yield. It's definitely the oil yield by the algae is much higher than the other vegetable non-edible oils which are reported. So these are certain scents which has a very high percentage of oil content. This I leave it for your reference. You can read it later. The only point that you need to keep it in mind is it is not only the yield. Or let us say you produce 10 grams of biomass, and out of it you check what is the yield. Okay, it talks about 25 to 75%. Again, this range is very huge. Again, you can't do anything with that. So you have to choose a species which gives a very closer range of oil content. So it is easy to handle and predict the oil quantity. So it is not only sufficient to talk about. There are 2 points people talk about. One is oil content, other one is oil yield. Both are very important. The microalgae. See, let us say the oil content of the species is very good, but it is the rate of growth is very slow. Then there is no point in when you multiply the yield with the oil content. Then overall thing has to be looked upon. So what needs to be looked upon is not only the oil content of the biomass which is produced, but it is also its growth factor, rate of growth. If the species, for example, when we started up reading about the algal technology and applied for a project, then even I also proposed for Botryococcus brunei as a species. But later I found it is very slow in growth. So if it is growing that much slow, then even the oil content is very high, it becomes useless. No? So you have to concentrate both on and also the given conditions. So what we worked is we also got some species from UTELs and other places, and when we tried growing that, it all failed. So the understanding was then whatever grown in the condition, that species we isolated and checked with the centers to find out what is the kind of species. Then we found it is Senidesmus. Senidesmus is a species which grows in Indian climatical conditions with a very good growth rate and good proportion, other than spirulina. Again, what is the knowledge gap for commercial possibility? So what are the points that need to be looked upon when you are going with the fuel to be produced from the microalgae? So it is basically, yes, definitely the strain selection. Strain selection, when I talk about strain selection, there are 2 things. It is not only the lipid content of the species, it is also the growth rate of the species. The species has to grow faster and also with a considerable amount of lipid content. Only then it becomes economically viable. Definitely it has to withstand the environmental conditions available outside. So you have to select a species for a given sunlight conditions, CO2 concentration and the water quality other than its lipid content and its growth rate. So you have to choose a strain in such a way that which matches with all these parameters, good parameters. Then you have to go with the cultivation and harvesting. So the cultivation, as said, it is you can think of going with the cultivation in two ways. One is open pond and a closed photobioreactor. If open pond reactors are fine, but when you see if there is an area of requirement is very huge, then you have to think of a reactor which will have a higher amount of surface area in smaller volume. So we also had applied for one patent with such a photobioreactor, which can be used in open sunlight. So with larger amount of surface area. Why open pond reactors are consuming large amount of space? Because it is having a limitation in depth. The depth of open pond reactors is only 30 centimeter. So the limitation is due to the light attenuation. So light cannot travel, even if you have a pure water also, within few centimeters light gets attenuated. You don't have a light intensity available at the depth. So if that is the case, then the algal cells are available, it is green in color. So the light attenuation happens within very few centimeters. So what is happening is, so you are limiting the depth of the open pond reactor. So we have also done kind of simulation work to optimize the number of paddle wheels used in the open pond reactor so that the mixing becomes better. So only if the liquid is properly getting mixed, then only the organ cells, all the cells get exposed to the sunlight, then it grows very good. So the cultivation has to happen in an economical manner using a right type of reactor, and then harvesting. So when we talk about harvesting, see, when you talk about closed photoreactor, the yield is very high. This amount of grams per liter is high. Whereas here the gram open pond is the grams per liter will be less. So for getting the same amount of yield or the biomass as an end product, the volume of water handled in the case of open pond reactor will be huge compared to a closed photoreactor. So as you increase the concentration of the species, the harvesting cost will come down. So it's an optimistic way you have to think that. So which type of reactor and what product that you are interested in. If you are talking about a food material or a pharmaceutical or a nutraceutical material to be derived from the algae, then it is only option. It is better to go with only closed photoreactor, where you also get a good concentration, so the harvesting cost comes down. If it's a fuel application or a biofertilizer or any other animal feed application, all these things are it is sufficient that you go with the open pond reactors. Then lipid induction strategies. So there are many literatures available which talk about nitrogen as a limiting source for producing more amount of lipid. So that is one strategy. So lipid injection does not stop only with the limitation of a nitrogen source. You can think of any other stress conditions given to the algae and check with the lipid whether it is getting improved. Let us say, for example, even mixing. You can think of increasing the mixing rate by which you are mixing the algal cells to get exposed to the light. There will be always some kind of mixing mechanism you will have in any reactor. Even the mixing speed can have an impact on the lipid production. So, or you think of light intensity, it may have an impact. Any stress conditions will have an impact on the biochemical composition which are given by which are produced by the algae. So you can vary the operating conditions, check the lipid content, then to optimize the strategy what is that will be adopted for enhancing a particular biochemical composition. Then, of course, the next is the cost-effective methods of lipid extraction and feed. So here, see, you get the waste product harvested by a energy-intensive process, and the air cross flotation is an energy-efficient process. And otherwise, you go with the centrifuge, then you get the wet biomass, then you go with the transesterification, then all these things. So instead, a dry powder, then you go with the lipid extraction. Instead, there are recent researchers, and one of my scholars is also working on how there is a possibility of extracting lipid from the wet biomass. So somewhere you have to think of getting the lipid or biodiesel with less energy input. So, and the other effect of catalyst again is there on the biodiesel production. And this is something like biorefinery concept, where we talk about more than one fuel to be produced instead of one. If you think of more number of products, always in any industry, byproduct. If you go with more and more number of byproducts being produced, the economy becomes better. And one more approach what we have also worked is you instead of thinking in biorefinery concept, they talk about more number of fuels itself being produced from the same biomass. Instead, what we worked is we have taken the lipid out. Then we tried using that residual biomass to be used as animal feed. So for which we have checked with the say once you have removed the lipid, then it will have protein and fats. So the protein content can be used by the animal as feed material. So then we checked the digestibility of the protein, and so instead of supplying that residual material as if it could be converted as something like a protein hydrolysate, then the digestibility of the protein becomes better. So, so there's a possibility. It is not necessary that you think of only producing more than one fuel from the biomass. It can even the other way around there. You can think of food and feed to be produced from the same biomass, one after another. So which sequence it has to be applied, all these things need to be worked out. So here again the bioethanol. Again as I said for lipid, there is a nitrogen content can enhance the lipid content. Nitrogen deficiency conditions enhances the lipid. Similarly for the carbohydrate also there are certain nutrient conditions and stress conditions enhances the carbohydrate yield. See, let us say if you are interested in producing ethanol, then you should think of increasing the carbohydrate content fixed by the algal species. Then the conversion becomes good. So we have also worked with different types of amino acids given as nitrogen source and checked with the carbohydrate content. There are certain amino acids which gives better protein than conventional nitrogen source, and carbohydrate is also good in certain amino acids compared to the conventional media used for spirulina. So spirulina we have taken all 18 amino acids and checked with different concentrations of what is available in industrial effluent organic amino acids concentration. We have checked with that and we have published an article in Algal Research. I will share that article maybe in the group so we can go through it. So which gives good interesting results. There are certain amino acids which also enhances the carbohydrate content. So if that is the case, that can be used for production of ethanol also. So here again the hydrogen production. Again this is again very minimum quantity of hydrogen is reported from the algae. We have not worked with the hydrogen production. This is a kind of mechanism. This I need to read. And here biohydrogen, again, as all of us know that it is cellular and friendly, all those things. Algae for biogas again. So we can think of what we said so far is algae to be used as a lipid or carbohydrate, lipid to biodiesel, carbohydrate to ethanol, and instead algae as a biomass because of its carbon content, hydrogen content, it can be used as a substrate for anaerobic digestion to produce biogas. So again, instead even it can also be thought about algae to be mixed with available waste material so that either the biogas content could be enhanced. The reason is all of us know that for anaerobic digestion process, the carbon to nitrogen ratio is very important. So you can think of you take the waste material, any waste material which you want to produce biogas through anaerobic digestion and mix it with algae to alter the carbon to nitrogen ratio so that the biogas content and the digestibility of the waste becomes better. So this can be thought of. It is kind of co-processing. It is possible that could be thought of being on a research level. So this is again a co-culture synergism. So we have also worked with algae to be used along with bacteria in wastewater treatment, and we had also published one article in RSC Advances. So this here actually it is shown what is the picture shows here is co-culture synergism for the biogas production. Same way it can also co-culture also can be tried with wastewater treatment also. Instead of anaerobic digestion, if you want to treat the pollutants, then along with the bacteria, whether the algae can work synergetic, can give a synergetic effect in treating a waste. So there again I have gone to a concept called microalgae assisted activated sludge process. Because many of you may know that activated sludge process is an efficient bioprocess which treats the effluent in a very good amount of BOD reduction and other things. So in that aspect, there are people also have studied on microalgae assisted activated sludge process. So we can think of algae to work along with the bacteria to treat the effluent or to produce biogas also. In that case, the algae will act as a substrate in a biogas. So here again, these are the things that are explained very well in the morning session. So I'm not going through in the detail. So only one thing that you can just see here, the paddle wheel it is generally only one side they use the paddle wheel. We worked on optimization of length to the width ratio and number of paddle wheels and the speed so that the flow profile and all we have checked this. And this is the cheaper mechanism of installation of algal cultivation. And you have other different types of reactors and where you have a pressure drop and other things, but the positive point is higher percentage of higher concentration and good environmental conditions. So that you don't have a problem of other microorganisms getting into the system because it's a closed system. That's an advantage. Whereas in open pond system, if the species is not robust, then you will get contamination in the pond. So here it talks about advantages, disadvantages. What are the things that I already said? You have a less productivity in open pond, higher productivity in the closed photobioreactor. This is again one of our research review articles that we have written on sustainable renewable energy reviews. We worked out what is the amount of CO2 produced per megawatt of power plant, then what could be the area and what are the issues, what has to be done to overcome the problems. So here only I said, let us say for 1 kilogram, 1 unit produces almost 1 kg of CO2. So per megawatt almost it becomes 8,760 tons per megawatt of CO2. So per megawatt of power, it produces 8,760 tons of CO2 into the atmosphere. So many hundreds, thousands of megawatts being produced, so many thousands, million tons of CO2 getting into the atmosphere. So just by calculation, so you approximately require 4.44 kilometers square per megawatt with the available production yield rate. So the instead of to reduce that area, the depth has to be increased. So that is where we worked on a photobioreactor with a higher depth, up to 1 meter of depth, so that the surface land area requirement becomes less. That's the only bottleneck when you go with the open pond system. By choosing a proper, by way of providing proper surface area to volume ratio, that problem could be overcome. So factors influencing photobioreactor design, as this is so the impact factor is now it's 15, close to more than 10. So it is factors affecting photobioreactor design, so species, surface area to volume ratio, light concentration of CO2. So these are the parameters which will affect. So here we have written in detail about the thing. And this is what I said, it is the influence of microalgal activated sludge process on wastewater treatment. So it is also reported, this is reported by the research paper, it is reported that a microalgal activated sludge process, the lag phase is reduced compared to only bacteria system. So it is faster. So this is these are the steps given that we can just go through it. And so here this is the point that I said. Industrial flue gases are used for it is absorbed by microalgae, then it is used for production of biofuels, so the CO2 becomes recycled. So more important is this slide you can just take up. And is it good to supply CO2 from an external source? Yes. See this reference talks about out of 432 identified metabolites produced by the algae, 37 metabolites showed significant changes between the elevated and ambient CO2 treatment. It means that if you supply a higher percentage of CO2 concentration for the growth of algae, it enhances certain metabolites. We have also checked with the Cellular species and Spirulina. When we externally supply CO2, the growth rate is increased. And there are certain pigmentation also were found different. It is good. So, in presence of elevated CO2 concentration, you get better advantages. This is what I would like to say. And this is another work that has been carried out in our lab on fish waste. See, in algae, the major nutrients, you have in media, you have major nutrients and minor nutrients. The major nutrients is the nitrogen, phosphorus, and potassium. So what we have done is we worked with nitrogen being replaced by the nitrogen received from the fish waste. We worked also with the prawn waste. Prawn waste, this kind of animal waste has got protein content in the waste material. So the prawn waste, the protein available in the prawn waste, it did not give a good yield. So we did not proceed our work with that. So we have taken the fish waste and we took a protein hydrolysate produced from the fish waste and we supplied that protein hydrolysate as the nitrogen source for the growth of algae. And we checked with what is the amount of yield, what is the quality of the protein, everything we have checked. It is all working very good. So what I would like to insist here is we have to think of utilizing the waste to produce from one place to be used as a raw material for this process. Then you are really your circular bioeconomy will work out. So what we have done here is we have taken the fish waste and produced protein hydrolysate from that, and we have supplied that protein hydrolysate instead of nitrogen source. So we have removed that the conventional nitrogen nitrate from the medium, and instead of that we supply the protein hydrolysate. So corresponding to that nitrate chemicals, what are the CO2 emitted that is saved into the atmosphere, and you are wasting the fish waste into the atmosphere, what are the emission gases it is going to produce into the atmosphere because of it is not getting treated. So that you are saving from the atmosphere. Emissions also you are saving. So you get two advantages. So we produced protein hydrolysate from the fish waste and supplied the protein hydrolysate at different concentrations. Then checked with the control nitrogen source. And I'll be just sharing the presentation, the results that we obtained. So fish waste we have taken, we have done the enzymatic hydrolysis, the biohydrolysis again. Then we obtained protein hydrolysate. This protein hydrolysate is used in Zaruk medium except the sodium nitrate. So sodium nitrate is removed. Instead we use only protein hydrolysate as the nitrogen source. Then the biomass is separated and analyzed for its properties. So what we have done here, recycled carbon to reduce the use of additional fossil fuel carbon. So here this is what I said, fossil fuel bio-based carbon. So these are the points that I had explained already. So here this is the sequence of steps what has been followed, but the point is whatever I said previously, we have taken protein hydrolysate and supplied that as a nitrogen source for the algal growth. The result is, I'll just share the results. Fish protein hydrolysate. This is the composition of the fish waste and fish protein hydrolysate. So we have analyzed what is the percentage of moisture and the fat, ash, sugar, all these things in the fish waste as well as in the protein, the hydrolysate which is produced. So this is the protein hydrolysate is used as the nitrogen source. So what does it indicate here is we have taken the liquid and we have directly supplied 3.3 ml per 100 ml of the medium. In that ratio, we have supplied the fish protein hydrolysate, and you can just check here the dry cell weight and productivity, how it is varying. So if you just check 0.5 percentage by volume, it gives a better yield of biomass, and we have gone with the biochemical composition analysis. We did not stop only with the yield. We also checked the biochemical composition of this biomass with the biomass cultivated or the algae cultivated in the conventional nitrogen source. And we found that the digestibility of the protein is better at a particular pH concentration than also the control. So here it is nitrogen assimilation rate. It is again good for 0.7. So here again, and we have also analyzed the chlorophyll carotenoid yield. If you just check this, a 0.5 is better pigmentation compared to the control nitrogen. So this is again good. And again phycocyanin, if you just check, phycocyanin is good at 0.5 pH concentration. So the point that I would like to insist here is either you go with additional amount of CO2 externally supplied and other nitrogen sources available from the waste, and there is also possibility you get an advantage of biochemical composition. It is not only helping the treatment of the waste, and also it helps to improve the biochemical composition of the biomass. So here this slide talks about the economy. What is the economy that you get? This is a reported value. It is we also go with the point. So that's why I'm presenting this slide. So if you take the fish waste, the meal for the animals, then the value that you get per kg is 0.3 to 1.2 euro per kg. Instead, you convert that to protein herbicide and use it. Then the value is improved. So on this, then again, if you go with this, let's say for a spirulina biomass, if you directly sell it, this is the cost, and you go with the derived products, and the cost may go up to even 4,000. So just check how many times it is improved. So I just given here this data only for you to make a note of it. If you go with the value-added products being derived from any biomass more and more, the overall economy can be boosted to a maximum extent. So this is the shed which is available in our department, where we go with the open cultivation, and these are the kind of reactors that we use. With this, I close my lecture. Any discussion we can have.
Thank you, madam. Thank you for this insightful lecture on algae. We had a good dose of algal biomass research from both Dr. Reena and yourself. I hope there's a lot that the participants have taken today on algal biomass. Thank you very much once again, madam.
Thank you. Thank you. Thank you, participants. Thank you.